Today, we're going to have two brilliant examples of initiatives showing how the implementation of Oxford Nanopore technology is driving discovery, whether it's to make headway in our understanding of the genetic of common complex disorder, or identifying and characterizing variants that were missed by standard method in individuals with rare disease. But first, I'm going to give you a very short introduction on our solution. So let's start at the beginning, the sample. Whether the focus is on rare disease in a clinical research setting, or if we are talking about a large cohort for complex disease, the most common type of starting material are blood, saliva, and buccal swab. But we also see tissue and cell analyzed even at scale. And of course, there's a growing interest in application using cfDNA. We have supported solution and experience with many of those, so please do not hesitate to contact us and inquire if you have any question about your specific requirement. And then once the sample is extracted, whatever the type of sample you are starting with, then it all becomes very simple and streamlined. So our PromethION suite of devices are perfectly suited for the analysis of human whole genome. In particular, our PromethION 24 with its A-series tower is a totally flexible platform enabling the user to independently run up to 24 flow cells and generate one human genome at 30x per flow cell. So what does it mean for you? Well, it allows you to run your sample to meet your timeline requirement. And that means if you want to run one sample at a time, you can do so without any batching penalty. But it's also ideal for production sequencing, generating almost 2,500 genome at 30x per year with a single device, which is almost double as what you could achieve with a review, for example. So if you look at a typical 5-day week, you can prep and start your sequencing run. And then by day 3, you can do your second prep and start your second sequencing run, which means you can do with one device 48 genome at 30x, and you can generate the FASTQ file and the BAM file in real time. And of course, the runs include methylation calling, both for 5MC and 5HMC. So you're generating genomic and epigenomic information simultaneously without the need for any treatment. And then when it comes to data analysis and interpretation, our unique variation workflow on Epi2Me desktop application offers a really comprehensive analysis, including obviously SNVs, structural variant, and again, methylation, as well as copy number variant and short tandem repeat expansion. Epi2Me provides secondary analysis, including annotation file and report for all the mentioned variant, as well as the SNV phasing block. Then the visualization of the map read and the different tracks are available within Epi2Me, but you can also open the output file in your preferred genome browser and do more of your custom analysis. And I'm thinking things like IGV. So now you can categorize all classes of variant that you need to investigate with a single workflow. Unlike most of the routinely used methods that leave gap, with our solution, you'll be able to get the same level of accuracy on SNP than with a NGS platform. You'll get indel, you'll get copy number, but with our unrestricted read lengths, our unrestricted read lengths are the only way to capture those 8% of the genome that are missed by short read and other methods. I'm thinking of complex structural variation, gene and pseudogen pairs, repeat expansion. And of course, again, you'll get a methylation profile at the haplotype level within every single run. So I hope I gave you a very high-level information on our solution. And I thank you very much, and I will let our excellent external speaker present.
Thanks so much for having me here today. I'm going to continue the story of undiagnosed individuals and how we can use some of these new technologies to get to answers for them. As you'll see, we don't have all the answers, but I do think we're making significant progress. So my conflicts here. You heard already in terms of thinking about some of the differences, similarities and differences, limitations of short read genome sequencing, especially compared to what we can get increasingly with long reads. You've already heard in many cases some of the advantages and disadvantages or differences, and I'll try and show you some of the differences between that. As we're doing this, I do think we're still not at 100% in terms of diagnosing those who sniff or who smell like they should have a diagnosis, but I think we're getting better, and we're starting to realize some of those regions of the genome that have been difficult to interrogate, some of which I have to admit I myself didn't even realize I was missing as we were going forward. And so I think as we do this, we're going to identify even whole new classes of genes, and especially, I'll just say one take-home point for me is that this methylation signature that you've been seeing about, I don't think we fully tapped in terms of being able to use that as a direction to point to where some of these answers are hiding. So within this, hopefully it's already obvious, but as we do the genome assemblies from short reads, it suffers from some limitations. If you have repetitive regions in particular, if you have regions where you've got duplications of the genome, there can be things hiding because your alignments really just aren't showing you what's there. You may have things that are essentially going in the garbage when you're doing the alignments, and so you're not recognizing what's there. Or if you have regions where you have very similar sequences, you may have such low read depth with the variant that you're seeing, it essentially looks invisible to you. So with this, you've got a lot of ability now because of the length of the reads to uniquely map these in the genome and again, be able to see things that we were missing before. You've already heard a lot about the epigenetic marks that we're seeing, some of the structural variants, as well as haplotyping or phasing. I'll say that in particular through some of the things that we had done through newborn screening, I wish I'd had more of that haplotyping and phasing to be able to see that for some of the recessive disorders. So I won't go through it again, but I will say this is, to me, was some of the most creative chemistry and technology when it first came out. I will timestamp it. It's been around a while. It's taken some time in terms of being able to develop, especially the analytical tools for this, but I do think we're on a trajectory being able to use this information more, and it really is just incredibly elegant chemistry to not have to do any amplification and to literally read the intact strand and to see those chemical changes as it's going through the pore and read out, therefore, essentially chemical modifications to the nucleic acid that you're reading. Just being able to do this in real time. For some of you, we haven't talked about it today, but there are even technologies to be in real time reading what you're seeing through the core, kick it out of the pore if you're not interested in reading it, and therefore being able to focus and interrogate your sequencing reads on one part of the genome that are really quite elegant. So as we're doing this and thinking about how to do this, there is the ability to flex in terms of doing relatively small numbers of samples, doing them relatively quickly, being able to do larger data sets and doing them with greater comparisons. I do think, as was stated earlier, the more that we have comparator data sets to be able to compare against, the more we will be able to recognize what's different about some of our cases. And so there are some of those large genome data sets now coming online, not just thousands genomes, but all of us and other data sets that are going to help in terms of that. I'm not going to go through in detail. You've already been hearing in terms of the pipelines to put your data through in terms of this. I will say these are in rapid evolution, and in a good way, I think are continuing to improve. As I said, especially as we think about some of the structural variants and some of those epigenetic marks. Within this, let me just point back. What I'm going to tell you about today is from Boston Children's Hospital, a cohort that's been, I'm a relative newcomer to the community, there for about 1.5 years, and I won't take any credit for it. But one of the wonderful things about the community is really the spirit of working together. And so one of the things we've created is the Children's Rare Disease Cohort, which takes then cohorts, several different cohorts collectively, but putting them in a common data pool where we can collectively work and help each other in terms of being able to diagnose those undiagnosed going forward. And that's proven to be extremely effective, and I'll show you about this today. One of the nice things that we've done at Children's, just in case it's helpful for any of you, is to have a common platform. So we fund it centrally in terms of having a common platform for any clinician to be able to use the tools that we build centrally. That way you don't have to be a genomicist in your own laboratory. We're centrally supporting that, constantly updating it as we're adding new tools, new ways of sequencing. Again, being able to democratize this for our clinicians, being able to take those individuals who know how to recognize something in the sequence they're seeing, but not necessarily necessarily do the huge informatics lift behind. All of this in essentially a browser that's very intuitive, hopefully, and able to see in terms of being able to browse this. And as I said, making it much easier, bringing more tools to more people is sort of the thinking in this. As we've done this, we did annotations that are similar to what many of you are used to looking at, and in doing this, trying to automate this system increasingly so that even as we're individually analyzing a case, we're surfacing the rare variants that are most likely to be phenotypic matches by pulling the current information from the EHR with the HPO terms and being able to use AI to then surface and prioritize those variants. So again, trying to think about doing this at scale and actually now in a systematic way going back and rechurning our data sets on a approximately annual basis. So what I'm going to tell you about today is a portion of the CRDC where we decided we were going to try and diagnose the undiagnosed. This was many of our investigators who came together. Shout outs to many of them. I'll just specifically point out Alan Beggs, who's been working on muscular dystrophy for decades. Alan has a unique cohort of individuals with true muscular dystrophy. He's done an incredible job in terms of phenotyping them. So the prior probability for essentially that entire cohort extremely high that there's going to be something genetic. I'll take the blame on the other side. I look at many congenital anomalies, and in many of those cases, I'll be honest, we don't really know the underlying genomic basis for those conditions. They may or may not be due to single gene conditions, and so you'll see some of the differences in terms of the diagnostic yield, but I put in some of our cohorts in terms of diaphragmatic hernia, esophageal atresia, and then we have many, of course, as many of you do, individuals with neurodevelopmental issues or neurological symptoms, and then just a potpourri of various different undiagnosed conditions. As you'll see, and this is true for many of us in terms of read depth, about 30x is sufficient in terms of the depth of coverage you need to be able to cover this, and the strand length approximately 15 kilobases. Many of these samples over a decade old. So just in terms of those of you who may have samples in your freezer to be able to go back to, still be able to dig back into those old samples and come up with some answers. So this is overall being able to see what happened with the cohort. I'll go in cohort by cohort and I'll show you some individual cases at the end. Still to say the majority, in other words, more than 50%, we still don't have a diagnosis. So just to level set in terms of that. On the other hand, it's pretty incredible how many diagnoses we did find reciprocally. So this is not just a onesie and twosie. There's really a large minority of individuals that we now have diagnoses and they really do fit. As we're going through I'll show you these in many different ways of slicing and dicing the data, but I do feel like what I'm showing you is the minimal in terms of the minimal yield. We were pretty conservative with the interpretations. So starting out, for instance, this is congenital myopathies and muscular dystrophies. This is, again, Alan Begg's cohort. This is the best of the yield, just to set expectations. Again, the prior in this case extremely high. Still, individuals who are unsolved at the top, but you can see a number of different, and I'll go back to this again, individuals due to large deletions, unmappable structural regions, areas where we were able to get phasing resolved. There might have been one mutation, as you'd heard before, in identifying both the second and the phasing, repeat expansions, and then complex structural rearrangements. And for any of you that are interested, the specific genes involved here, and I'll go back to a couple of those cases. As I was alluding to before, for diaphragmatic hernia, the yield not as high, likely because the complexity in terms of the genetic architecture of more complex. Some of these due to single nucleotide variants, others due to more complex structural rearrangements or complex indels. Esophageal atresia, again, within this, still the majority not yet diagnosed. Again, the underlying complexity for many congenital anomalies, probably more complicated. Certainly CHD7, a well-known gene for many of you in terms of esophageal atresia, hit recurrently, as well as some complex structural variants. For those with neurological disorders, again, the majority still not solved, but perhaps not surprisingly, a repeat expansion as well as single nucleotide variants. And then I'll highlight one of these cases, but again, structural variants that we're seeing as well as duplications. So overall, in terms of the cohort, for those of you who want to be keeping track in terms of what you're seeing, still, again, we see a variety of different types of conditions, different types of variants, that is, that perhaps weren't seen on short read sequencing. As we go through and look at this and look at the yields by the different cohorts, this is what I meant by that it's still the minority, but overall it's now a quarter of the cases, 24% of the cases where we didn't have a diagnosis before, that with this technology we're able to see and call things that we weren't able to before. So not bad for me in terms of, I don't know about you, but the families that are coming back to me year after year in terms of what are you going to do for me lately? These are the things that we're trying now in terms of solving those cases. So for the ones that we're trying to think of in terms of where is long read really uncovering and giving us additional value, some of these cases are highlighted here. And again, as we think about this, they're cases that have the methylation patterns are giving us, I would argue, an orthogonal way of reading this out. It's essentially a functional view of the genome in addition to what we might see from some of the variants. Makes me increasingly confident that what we're seeing is really causative, as well as I'll get to in a little bit, some of the more complex structural variants that we're seeing. These are the ones that I think the long read technology, there's no way. In going back and looking at our short read sequencing, standing on my head, doing other things, there's no way in the world I would have picked this up from what we did before. And those are things where we didn't have parents to be able to get the phasing resolved. We had issues in terms of especially the repeats, some of these complex regions of the genome, and I'll show you one of the case where you've literally got duplications and you can't uniquely map them back to be able to see. These were things that we could not have been able to see based on our previous data that we had. Within this, I will also admit that some of these things we did see when we went back to some of our old short read data. But for all of us, it's amazing what a tincture of time will do. That is that the same, even if it were as little as 2 years ago, the same variants that I saw with new ways of being able, new genes that have been identified. And I will say personally for things like alpha missense in terms of being able to prioritize different variants, especially missense variants, I think we're just able to recognize much better. So part of this is admittedly the technology. There's another part of it, which is our knowledge base and our general ability to interpret variants over time. So a combination of the two of those were incredibly powerful. So let me relatively quickly go through 3 cases just as examples. This was a little girl who came to see me when she was a baby with overgrowth, and in doing so, really didn't have particularly distinguishing features. I thought she might have Beckwith-Wiedemann syndrome when I first saw her. We had done the usual BWS, and you've seen some of those assays already, and didn't come up with an answer, but she still bugged me that there was something that was going on. We went through and did her sequencing here, and I'm sorry, my pointer's not going to work very well here, but she's on the top, and you can see the read depth coming down in the middle there, and that's the region where she has a deletion. Her mother and her father on the bottom. You can see that they have, this is a de novo deletion within that region. They have normal read depth. This particular region was in the 3' untranslated region. So whether or not it was actually functional was not clear to me. So this is where I bring in the methylation pattern as being a good readout. As you've seen before, the red being areas that are hypermethylated, the blue areas that are unmethylated or less methylated. And again, I wish my pointer were working here. In the middle here, you can see what's called haplotype-specific hypermethylation. And what you'll see is that for her reads, you can see more red than blue in terms of being able to see the regions that are hypermethylated. And in that particular case, hypermethylation actually increases expression of this gene. So she has overexpression of this gene. Why does that matter? Well, there's a condition for this particular transcription factor. It's interesting that there's a sort of Goldilocks in terms of being too hot or too cold. So it can be reciprocally associated with something that looks like Silver-Russell syndrome in terms of lower growth, or on the other end, Beckwith-Wiedemann syndrome in terms of on the higher side of growth. And in particular, although I'd seen her as a baby, as I got these results, called up her mom to see how she'd been doing, and she said, you know, the funniest thing is her teeth. When she was a baby and saw you, she never had this tooth issue, except now she has so many extra teeth that her dentists don't know what to do. And in fact, that was the clincher for me, because that is a very, very specific finding that's associated with this particular gain of function or overgrowth for this gene. So that's just one example of what this can do, but again, something that we hadn't seen before and hard to know what to do sometimes for these regions that may be in regulatory areas. This was one of Alan's cases that was an 18-year-old, which on muscle biopsy clearly had muscular dystrophy. In this particular case, he knew one mutation from short read sequencing, trying to figure out whether or not there was a second mutation that might have been missed. And so you can see on the right over here, the region where there's a little bit of a gap. You can see a deletion of a few nucleotides, and then on the left in the areas of blue, a complex duplication. Now being able to put these together and see whether why we didn't see this before, and I have to admit this was one of these new things to me. This gene has a region where it's several exons that are entirely duplicated. So it's not just one exon, but it's a chunk of exons that are duplicated over 3 times within the gene. And so if you have a mutation in one of those exons, as you pile those together with short read sequencing, it basically gets lost in the noise. You don't see and you don't uniquely map. On the other hand, as you have the long read sequencing for this, you can actually see each of the strands individually and you can read them out and you can clearly see these regions. So there are regions like this I'm sure that many of us are just not used to looking at. If you now put these two together in terms of being able to understand the haplotype, you can clearly see the 2 different types of mutation and you can clearly clinch the diagnosis in this particular case. Finally, I'll just say that this is one other little girl with a myopathic process that wasn't clearly defined. This is, again, a region where this particular gene, Notch2NLC, can be associated with a repeat expansion. She is shown in the middle here. The regions that are purplish on the left there are the repeats, and the number of those repeats are what are shown in the box. You can actually see, like an accordion, that each of her alleles has different numbers of repeats, so there's also probably some somatic instability with this. None of those repeats on her father's side on the bottom, and a smaller number of repeats for her mother. And then you can see, again, this beautiful functional readout in terms of looking at the methylation pattern with that red showing, again, that she's hypermethylated in the region, unlike her mother, and probably the reason why she appears to have something that is not seen in the rest of her family. So just to briefly wrap up, in doing this, it is, not perfect. No technology is, but it's starting to show us and help us recognize things that we haven't seen before. My prediction is that we're increasingly going to use this. You've seen how it's being used on the UDN or used in more in a research phase, but I won't be surprised if some of you, possibly laboratorians, are looking at this, that you may start incorporating this more in terms of diagnostics as we go forward. Most importantly, just to thank all of the incredible people who've been part of this. I just get to be up here and be able to let you know about it. So thank you.
Cassie and I are super excited to show you how the NIHR BioResource is using Oxford Nanopore technology to transform health research. But first of all, please note the disclaimer from ONT that their product is only for research use only, and also that we have no conflict of interest. So we'll spend the next 15 to 20 minutes first to introduce what is the NIHR BioResource and how we have set up a high-throughput pipeline for long-read sequencing, and then share with you some of our preliminary results as well. So the NIHR BioResource is one of the 4 pillars in the UK genomic landscape. It's all about bringing people together and leading research. But the aim is always for the health benefits as well as innovation. So to date, we have about 350,000 participants on our panel. But our unique selling point is the recallability of our participant, either by genotype or phenotype. So every participant will give us some information about their lifestyle, about their health. They will also give us biological samples. And with that, obviously, we can do genomics. So we have this first stage in the BioResource of banking information. But actually, the aim is always to match those researchers, those participants for innovation, for health benefit. And the second, maybe, unique selling points of the BioResource is that actually the panel is based on sizable cohorts of participants with health condition, and also those who are, in general, very much underrepresented in health research. So we will have some control healthy general population, and this is through our collaboration with NHS Blood and Transplant. But we have participants with conditions from bowel, mental health, liver, inflammatory conditions, rare diseases. During COVID, we obviously created a COVID BioResource, making sure that we were doing some good work for this time. And more recently, our activities have actually reached out more to those who are underrepresented. And by these, I mean the young individuals, young people who are never part of research in general, and also individuals from Black communities. And those are our Decipher and IBO programs as well. So what about long-read sequencing? Well, it all started for us as well with rare diseases. When the BioResource wanted to actually make a challenge, it was a challenge, make a difference for rare disease patients. But actually, I will leave you with Kathy, because she will tell you more about this.
Thanks for the introduction, Nathalie. So yes, my journey with the BioResource started with this big sequencing project. We were aiming to sequence 8,000 genomes ourselves, and we worked with Genomics England and did 5,000 of their pilot genomes as well. What we really took home after the analysis had finished, we had a Nature paper I've referenced there, but also at least 50 other papers came out on all sorts of aspects of rare diseases and technology, is that we only got a diagnosis for 20% of the patients that were part of the program. Other studies around the world have had similar findings, and we want to know why. We want to do better for these patients, and that's where our journey into to long-read sequencing has started. So our long-read project, it's always we always try and push the boundaries a bit, so we are trying to do up to 22,000 long-read genomes, and there are 3 projects involved in this. There's our rare diseases one that started it all, but we've also working with eating disorders, and we have a general population cohort in there as well. It's obviously all Oxford Nanopore. We're using Prometheans, and the lab work is a collaboration between our lab in Cambridge and one in King's College London. All the data is then processed at the University of Cambridge High Performance Computing Facility, and then following our initial publications, this data will be made available through a data access process for the international research community. So the 3 sub-projects we've got going on, one is related to genes and cognition. It's a dementia-related experiment. We have cognitive profiling data on over 30,000 participants within the BioResource across all those different themes that Nathalie mentioned. All the participants were sent an email and asked, would you like to take part in this app and get your cognitive profile? And we are going to be sequencing 14,000 of those. The majority of those will have 2 time points in that app. And the aim is to look at structural variants and methylation status that might improve understanding of this precognitive decline. We've also got a 4,000 participant eating disorder sub-study, and that's looking at the 2 different extremes of eating disorders. So it'll be about 2,000 people with anorexia, so low BMIs, and 2,000 with binge eating disorders, so really high BMIs. And this is a really underserved population. There are no real drugs to treat these types of diseases, and we're hoping that the research in this area will be really beneficial to these patients. And then finally, the rare disease part of this is 4,000 people where they have had previous genetic sequencing that has not been able to give them a diagnosis, but there will be some cohorts, including family members, and some trio analysis going on. And the aim is discovery, but also, where possible, to get research diagnostic reports back into the NHS for verification, to give answers to these families and end their diagnostic odyssey. So it's been quite a journey from piloting these processes in our labs. We have 2 laboratories working on this using 8 Prometheans. So we've got pictures of both of the labs. King's has got 6 Prometheans. You can see them all in a row there. And our Cambridge labs are a little bit more modest with 2 Prometheans. We have both blood and saliva types. You'll be seeing some data on that in a moment. And we have some really quite fresh samples and some legacy samples. So real-world samples, I've heard them described recently. We're aiming for 30x genomes. I'll explain why in a moment. We're doing the variant calling and alignment on the boxes. With a wash and reload strategy, which means that we can do 24 genomes per Promethean every 72 hours. So between those 8 machines, we can do 384 whole genomes every week, 1,500 every month, and up to about 18,000 every year. So that's how we're aiming to deliver this project. Then we have our bioinformatics team doing all the downstream analyses. So just very quickly, that's the bioinformatic workflow. We've got a lot of work going on those Promethean towers that Gordon was showing, how many calculations they can do a second. Well, they're doing a lot of the hard work with the base calling and alignment, but then the work continues at our high-performance cluster at the University of Cambridge, looking at the 5 main variant types and doing all the extra pipeline work there. Beyond that, we also do, we're setting up a lot of QC analysis to make sure that the participants we think we're sequencing are the participants that actually got sequenced, that there have been no mistakes in the lab. We're also looking at cross-contamination between samples, or making sure that there isn't any of that. So there's sex checks, we check the phenotypes, heterozygosity, we're looking at the ancestry, which factors into the downstream analyses, but we're also looking at the quality of the sequence that we generate, so looking at missingness of data, Hardy-Weinberg, allele frequencies, things like that. So I've just put a quick picture in there. I always like a PCA, so this is the ancestry analysis. The coloured dots are all from the UK Biobank GWAS, and then the black dots are our first 1,500 people, and you can see they overlay really nicely onto that analysis, even though they are different data types. So we are about 6,000 genomes in, but because the bioinformatics takes a little bit longer and we had to get our slides ready for London calling, this is a snapshot of the first 1,500. They're spread over the 3 different cohorts I mentioned. You can see that our cohorts, because they are UK cohorts, they are tending to be very Caucasian biased. We deliberately skewed some of these early samples in the genes and cognition cohort to be some of our more diverse genomes in the bioresource. So that skew will go away as we fill in the 14,000 samples there. But you can see that we've got a good range of ethnicities and genders, and you can also see the eating disorders cohort is actually heavily female skewed. Even in that first small subset of people we've started, we found one person XXY in there as well. The other complication we have with these real-world samples is we use both blood and saliva samples. So any plots coming up, if the plots are in blue, that means there are saliva samples, and if they're in a pinky red, there are blood samples. We also, because these have been banked over 15 years, a variety of different extraction methodologies have been used when generating these DNAs. I'm not saying anything about you should use one technique over another. These are just what we happen to have to use. The DNA is being extracted, we have to cope with those, and we do see differences in the method or because of the age of the samples. So that's just a quick spread of the different methods we've used. Here's a first look at the data. So the N50s for blood do tend to be a little bit higher than the N50s we get for the saliva samples. They are a little bit more degraded, harder to work with. The depth of coverage, we are aiming for 30x genomes in all the cases. It's a little harder to manage with some of the DNA inputs, particularly the saliva ones, so there is a broad range there. And the Q scores generally all high 18s or 19s and above, so nice Q scores coming out of this analysis. This is the problem with working with saliva. It won't be a surprise to you. Blood, of course, lovely to work with pretty much every read mapping to the human genome, but the saliva samples obviously include microbes and sometimes people's lunch. So some of those samples are really good. They do map really well to the human genome, and others you can see less than 50% of them are mapping. So we do have to over-sequence some of these saliva samples to get to the 30x coverage. So they are a lot more challenging to work with. We did do some benchmarking with some of the genome in a bottle samples. So this is a slide summarizing 6 samples we ran, 3 in Cambridge, 3 in London lab, to make sure that the labs were producing comparable data. And you can see that the F1, the precision and the recall are all working out really nicely when we run the samples. The reason we want 30x coverage is you can see that the SNV plot there is starting to plateau at around 26, 27x. So we think we're calling all the variation in the SNPs there by 27x. At similar depth of coverage, it's about 0.95 for the structural variant. So the more coverage we get, the better it is for the structural variants, but we have to work with the samples that we've got, and it's just not always possible to get above 30x. Looking at the count distributions across the different variant types. So we've been looking at all the types, structural variants, SNVs, indels, deletions, and the methylation there. You can see that across all those 5 sample types that we've been trying to optimize, the numbers come out pretty similar. A little bit less in some of the cases for the saliva samples, but as we've explained, the coverage is a little bit harder to get for all the saliva samples. And you can see there's another slide coming up, but the skew in the Quiogen samples is because of those non-Caucasian samples that we've deliberately put in there. So we're quite pleased to see the consistency across all the variant types with our data set. One interesting and pretty recent observation, so we're still looking into this, is on the methylation. And we've been finding that the pass rates do vary a little bit in the saliva data set. They seem more consistent in the blood data set. So there is a correlation between the pass rate and the coverage in our saliva samples that may well relate to this lower confidence in call rate when we have a lower coverage. So it could be that we need to do as best we can with the coverage to get the best methylation data out of our saliva samples, but we'll keep looking into that. This is the picture with the ethnicities, so it's no surprise that the SNPs and structural variants are more prevalent in our African and Middle Eastern populations within the bioresource, but the methylation signals are pretty consistent across all the populations. And then the other thing we wanted to do was prove that the technology was working as well as the previous technology we'd used. So as we said, we'd done a lot of work on rare diseases. So we went back to some of our rare diseases where we had confirmed cases and fed them back to the NHS and had them validated by the NHS. And we wanted to make sure that this technology would pick up all of those variants as well as all the other extras it adds. So I've got a couple of slides coming up just looking at a SNP, a de novo SNP in a trio. It's an example from one of our pulmonary arterial hypertension rare disease cohorts. Got 2 examples of compound heterozygotes. They both happen to be in our primary immune disorders rare disease cohort. And it's really nice seeing this in the nanopore data, how compound hets can work, and that does not need to have a trio to determine this. And we've also got some examples of some structural variants that we found in our retinitis pigmentosa, or one of our eye disorder cohorts, in our bleeding and platelet disorders cohort, and another one in pulmonary arterial hypertension. So the first one's fairly standard. It's at the top of the graphs, you're looking at the original short read data, and then the bottom of the graphs is the trios with the long read data, and usually with phasing turned on. So if this was a SNP, it's only present in the proband. That's why it's a de novo cause. So that was simple. The next ones are compound hets. In these cases, the 2 variants were in genes either 8,000 bases apart, or one of them is 25,000 bases apart. And so this shows the power of long-read sequencing to have those long fragments so that you can phase the genomes. So in this example, you've got the whole trio. One of the variants comes from the mother, one comes from the father, and in the proband you can see that one chromosome has the mother's variant, the other chromosome has the father's variant, and that means both chromosomes, both copies of the gene have been knocked out, basically, which is why the person has the phenotype. And normally with short read sequencing, you need to sequence the whole trio to understand that. With long read, we could get away now with just sequencing the proband. And this is often really important to us because we don't always have the parents available or other family members in order to do that. So being able to do phasing is really exciting. And I know our primary immune disorders cohort very excited to look at more of these. So this one's just another example. In this case, we had a mother and a brother rather than a father to do the phasing with. And then the final ones are some deletions. So this one's a large homozygous deletion. So you can see the breakpoints come out really, really nicely there, and that both chromosomes are basically taken out by this deletion, which causes the phenotype. This one was a deletion and an SNV, so you should have zoomed in a little bit more, but there is a heterozygous deletion in there that matches what we found previously. This one is another large deletion that takes out the 5' end of the gene, also explaining the phenotype. And then finally, the other thing we're really interested in is methylation. That's another really powerful aspect of this technology. So this is an example from one of the genome in a bottle samples looking at PEG3, which is well known for imprinting. And you can see in that bottom trace there how the lines diverge, and that one haplotype is heavily methylated and one haplotype is not methylated. And this is exactly the sort of signal that we're hoping to see in other genes throughout the cohorts that we're looking for. And hopefully explaining some other phenotypes. So, in summary, here at BioResource, we're building this resource of up to 22,000 human genomes, all based on the UK population. We've demonstrated that we have a workflow that can scale to 48 human genomes per week per Promethean, with 30x coverage. We've shown you an example of the sort of data quality that we're obtaining, both using diverse sample types, so that's blood and saliva, old and new samples, and also different participant demographics. We've demonstrated that we confirm all the diagnostic type variants and methylation signals that we would expect to see for the analysis that we want to do moving forward. So in summary, BioResource is actively enhancing multiple health themes with rich genomics datasets, and we're hoping to revolutionize gene discovery and diagnosis rates and treatments and clinical care for patients all around the world. So thank you for listening. There's some photos of the lab team, the bioinformatics team, who are also really key to this. We're a bit too shy to give me pictures. Thank you to all of them, to all of the BioResource, without whom we wouldn't be able to do this work, our funders, and of course all the BioResource participants. If you want to know more about us, we've got emails, we've got Twitter, or X as it's called now, LinkedIn, we've got a webpage. Do get in touch with us. Thank you very much for listening.